Hi, my name is Natalia Thomas, and I will be presenting my pilot study investigating the effects of sugar alternatives on U87, human-derived glioblastoma, cell proliferation, and survival. I conducted this experiment as a follow-up to the increasing number of clinical trials utilizing the ketogenic diet as an adjuvant therapy for glioblastoma, a stage 4 brain cancer. The ketogenic diet is a high-fat, medium-protein, low-carbohydrate diet that targets tumors by decreasing the glucose availability needed for cancer cell survival via aerobic glycolysis. Mouse models with induced glioblastoma tumors have found a significant decrease in tumor metastasis following a ketogenic diet as opposed to a standard diet. People on the ketogenic diet typically look to sugar alternatives to replace their favorite sugar-containing foods. Although no association between sugar alternatives and carcinogenicity exists, examining the effects of sugar alternatives on already existing cancer cells is important when considering the effects of using a ketogenic diet as a treatment adjuvant. Thus, this experiment seeks to question the effect of artificial sweeteners on glioblastoma. To investigate, U87 cells were treated with various sugar alternatives for a week. Erythritol has been shown to enhance U87 growth at physiological concentrations. My hypothesis is erythrophosphate is converted from erythritol and serves as a metabolite connection to glycolysis. If this hypothesis were to be true, then increased cell growth should also follow aspartame-treated cells. My experiment consisted of seeding 15,000 cells per well in a 6-well plate, treating them with 25 millimolar of allulose, saccharin, sucralose, erythritol, aspartame, or stevia, and assessing their proliferation survival via hemocytometry after 8 days. Due to issues with controls, the results were inconclusive. Optimizations are currently being put into practice to achieve an effective protocol. The major issue among the list was that of a low density of seeded cells, which was initially done to compensate for the long period of growth. To mitigate this, cells will be seeded at 50,000 cells per well, and the length of time for the treatment will be shortened to 4 days. Once an established protocol can be created for this experiment, future interests include looking at concentration dependence and physiological concentrations, as well as a mouse model that will look at a standard diet group, a ketogenic diet group, a keto group plus sugar alternatives, and a standard diet plus sugar alternatives. Overall, despite lack of conclusive results, this pilot was full of information that can be used to avoid error in future experiments. Thank you.